Right, this is our first lecture in our Unit 2. This is going to cover Chapter 7 in your textbook, membranes. And just hold on one second. Yeah, there we go. All right, Jack's going to come in and say hi.
Hi.
That's his arm. There he is.
Hi.
That's Jack. And of course, he can't hear you, so don't. But
Mm-mm.
You guys have
anyway,
the same glasses.
yeah, they said we have the same glasses.
Oh,
Now he's awkward. You didn't. Okay.
I'm missing.
You want to go? Go. Go. Have fun.
Thank you. Bye.
I think he said bye to you guys. That wasn't to me. All right, here we go. Here we go. I'm gonna watch this 20 years from now. Like, oh, look how young he was. So this membrane, this is the third functional area of a cell, right? We focused last chapter on chapter 6. We focused on the inside of the cell. We talked about the cytoplasmic components. We talked about the nucleus. Now we're going to talk about the membrane. And this is arguably the most important part of the cell because this is the boundary of life, right? So what the membrane does is it selectively allows some things to come in and some things to go out, and it traps some things within, and it prevents some things from getting in, right? So the idea here is we have an internal environment that is different from the outside. And we're going to establish that difference, and we're going to maintain that difference. And that's what homeostasis really is at the cellular level. Establishing and maintaining, you can think of them as gradients, concentration gradients. And we talked about this a little bit in the last chapter, at the beginning of it, the idea of cell size. So this is here just to review. The idea that cells are small because they have to continually interact with their environment. And the only place they can do that interaction is at the membrane. Yes, there is supposed to be something on the screen. There it is. I thought I was sharing my screen. Let's scroll back here. All right, so you guys are just listening to the sound of my voice, looking at darkness. Okay, so let's start. Here we go. So there's a membrane, okay, and you can see there's the external environment and the internal environment. And so the idea is some things are allowed in and some things are allowed out. And some things are trapped in and some things are trapped out. Right? And so in this game here, which requires energy, this is the maintenance of the homeostasis, right? So maintaining homeostasis means keeping that internal environment different from the outside, which makes sense, right? Because if the inside and the outside of the environment were all the same, then there'd be nothing there. There's no life. You can start to understand like what life actually is now. You can't really define it still, but one of the things that makes life life is this idea of a pattern, establishing and maintaining a pattern of some kind that's distinct from the surroundings, like an ocean wave. And then we review this concept of cell size as a reminder. We talked about this at the beginning of chapter 6, this idea that cells are small because they need to be able to quickly interact with their environment. And they can only do this at the membrane. All right. We talked about how cells are built. In particular, we're going to use eukaryotic cells as a model for our discussions because eukaryotes are the ones that this entire class focuses on, as opposed to like a microbiology class. And the reason why the eukaryotic cell is so big, remember, is because of all of these internal membranes, right? So if you just picked a random spot on the screen there, you're never too far away from a membrane. You're never too far away from the ability to um, move something in or out of something. So there's all of these environments that we've established. And let's also review and remind ourselves of the phospholipid itself. This is back from chapter 4 now, chapter 5. The idea of this amphiphilic or amphipathic molecule that has both the polar head and the hydrophobic tails that allows us to establish a gradient, allows us to establish a membrane to begin with. Right? So we have the phospholipid and all the different kinds of phospholipids. And the way they interact. Remember, there's the inner leaflet and the outer leaflet, right? So that's going to be our starting point here. And the fact that there's cholesterol on the membrane, remember the cholesterol stabilizes the membrane. We talked a little bit about that. We'll talk more about that today. And the fact that when you put these phospholipids into water, depending on the temperature and the pH and the lipid concentration and the ionic concentrations, they can form these different types of structures. All right, now this is a slide. It's all text. Like I hate all text slides, but this is a slide you could review. This establishes the rules of the game. And so I'm gonna skip this slide, use this for your study. I think this is in the study guide too. But instead what I wanna do is I wanna go to the whiteboard now. And so I have to share my whiteboard screen. And this is where I wanna do a little bit of active learning for you. Because the one thing I want you to get out of this lecture right now is the rules of the game, so to speak. I really have to reboot my computer. I can't write like this. Oh my gosh. Hold on. Just want to zoom in around to something. Yeah, around here is where. But my Wacom graphic driver, that's why. So this is me trying to draw a circle. I can't draw circles. Okay, just give me 1 second. I'm gonna try to shut down, doing the Control Delete, and there it is. Anything with it, Wacom, and then please work. Please, tablet driver is not responding. Turn off your computer and turn it on again. Never mind. I'm not. Never mind. I can do this with a broken tablet. So rules. Just there's two variables: charge and size. Okay, so this is me teaching you chess. This is me teaching you basic rules, and then we're going to apply these rules to problems. Okay. So rule 1: big things can't get in the cell. That's obvious. That's the easy rule, right? It just makes sense. Big things can't get in. It can't just leak into the cell. Remember that the membrane, being this phospholipid bilayer, right? This isn't a solid. This isn't like a protein coat, okay? This is like more like an oil layer. So some things, so big things will physically just not be able to get through that. Little things, okay, little things that are uncharged, that are non-polar, they can literally pass through the membrane like it's not even there. Things like oxygen gas or carbon dioxide gas, the gas exchange, you breathing in and out right now, all that oxygen gas you're breathing in and all the carbon dioxide you're breathing out are passing through membranes like they're not even there. If you zoom all the way down into your lungs and you look at the alveoli and you look at the little cells in there, the oxygen and the carbon dioxide are just diffusing right across that membrane. Okay.
You said if they're non-polar?
Non-polar. That's the key here. non-polar. So what is that? So here's my first little active learning. I want you guys to take what I gave you two simple concepts. If they're big, they can't get in. If they're small and non-charged, they can get in. Okay, so what's the relationship between those concepts and a charged molecule? Only non-polar gas, no, so not just gases, but solids too, non-polar solid. So they're small non-polar molecules. So for example, hydrocarbons, small hydrocarbon chains. This is why if you've ever gone and pumped your own gas and you've accidentally got gasoline on your hand, you're like, oh, that smells terrible. And you go in, you try to wash it off, and you wash your hands, and you got soap and water, and you're done with it all, and oh, it still smells terrible. That's because the gasoline actually went right through your skin. You can't wash it off. It's inside you because it's lipid soluble, because your skin is made of cells, and cells have a membrane, and small non-polar molecules easily pass through those membranes. This is why gasoline is dangerous. That's why in New Jersey they banned you from pumping your own gas. Instead, they make all the workers get exposed to the carcinogens. That's more ethical somehow. Right? Don't let those people poison themselves. You poison yourself for them, and we'll pay you a little bit of money for that. Not too much though.
So it's not good when it gets on you.
Right. Now, that said, a little bit of gas on your hands every once in a while is fine. If you're working with gas every day and you get it on your hands every day, that's not fine. Okay, so that's a good rule of thumb for your life, for working in lab. If you get a little bit of something on your hands, depending on what it is, I mean, there's some things that even a little bit of it will do some permanent damage. But the vast, like a little bit of virus, never hurt anyone, right? Wait, no, that's not true at all. that's so very wrong. No, but most things in the lab, you're not going to do permanent harm to yourself if just a little bit of it gets on your hands. And you can pump your own gas if you get a little on your hands. You don't wear rubber gloves when you're using the gas pump. You don't have to go OCD on me here, right? But if you're in the garage and you're pouring gas in your lawnmower every week and you're spilling it all over the place, first of all, you're doing something wrong. You should probably learn how to pour gas in your lawnmower. But if you're getting that all over your hands every week, that's not good, right? Or if your dad's in the garage every week doing that, that's not good for him either. And he's by this point probably been doing that for years, so you might want to have him stop doing that. So what I want you to do now is think of the game. There's things outside the cell that can get in, and there's things outside the cell that can't get in. What are they? Okay, so let me throw something. How about a protein like insulin? Can insulin just pass through the membrane? What do you think?
No.
Good. Too big. All right. What if I took insulin and I made it completely electrically neutral? How about now?
But it's still too big.
Good. Right. So the most important variable is how big things are. Okay. Now it's small. Then the next question you're asking yourself is, well, is it charged or not? How about polar molecules? What about polar molecules? So Ariana, you're saying no. But what's your thought process there?
So don't they have to be non-polar?
Right, because I said that, because I said so. Right, but so instead of just following the strict rule there, why can't polar things get across the membrane?
Wouldn't it mess up like the charge of what's already been established?
When you say mess up the charge of what?
It
Of the
would-
molecule or the cell?
Of the cell.
Okay, so by mess up, what do you mean?
Well, if it was already a specific charge and you're adding new things with opposite charges, it's going to change.
Oh, okay. I like that idea, but here's the truth of it. The insides of, I want to say all, that's a dangerous word, but the insides of all eukaryotic cells. Okay, so if this is a eukaryotic cell, the inside of the cell tends to be more negative than the outside. And the reason for that is there's a pump that we'll talk about, the sodium potassium pump. And what the sodium potassium pump does is it establishes and maintains that electrical gradient. Okay, so there's an electrical gradient that cells establish. Right, so now you can imagine if the inside of the cell is negative, and we have this polar molecule here, can you imagine that as it gets a little closer to this, if its polarity is also a little negative, it's going to act kind of like a magnet pushing away from another magnet, right? It's going to literally get pushed away. If this is slightly negative, right? It's just, there's going to be a little bit of a repulsion, not too much, but a little bit. Likewise, if it's a little bit positive, what do you think is going to happen then? Would it go in? Remember that the heads of phospholipids are polar. So what's going to happen to a positively charged thing? Can those go in?
Those just be attracted to the negatively charged of the phosphate.
Excellent. So in other words, positive charged things are going to stick to the membrane. Let's use red so you can see this better. They're going to stick to the membrane. They're not going in either. Good, Zach. And it turns out, as we'll see later in the semester, remember H+, remember protons and pH and all that? So protons will actually accumulate along the surface of a membrane. Okay, so here's the rule. Charged molecules and polar molecules do not get into the cell. They do not pass a membrane. A membrane is impermeable to them. Okay, so the easiest way to think of all this is to think of the one case of things that can get in and commit that to memory, and then you can intuit or logic your way to everything else. And that is small, uncharged particles can freely go across the membrane. The membrane is permeable to non-polar small molecules. What about medium-sized molecules?
Hey, Dad.
Hold on a second. Yeah, what's up, buddy?
Am I allowed to watch TV?
No. He knew the answer to that before he asked. He just wanted to make me look bad in front of you guys. That's kind of like, remember when you were little and you're like, hey, mommy, can Bobby eat dinner over tonight? And like you ask in front of Bobby, and if mom says no, then she's a jerk. And so, of course, she has to say yes. That's what he just did. But I don't care. I can look like a jerk. He still can't watch TV. They watch so much TV lately. Anyway, right, Jack?
What?
He said what? I thought he might have been eavesdropping. That's just paranoia. That's just paranoia. All right. So right, you got the rules of the game. That's it. It's that simple. All right. So now here's my next question to you. Well, how do we get things that are impermeable across the membrane? What do you think?
Energy.
Okay. So like brute force, just shove it in there. Just
Or-
push it really hard until it, I mean, I guess that I'm sure there's some mechanism somewhere in biology that just brute forces it in there. But so like I like that. They're like energy. Okay, yeah. But so what are we gonna do with that energy? Right. I'm not really sure here. All right. Well, so here, I'm going to give you a little hint. Remember when we learned about the rough ER? What was special about the rough endoplasmic reticulum? What did it do?
It had ribosomes on them.
Okay. And what did the ribosomes do?
They produce protein.
Good. And what kinds of proteins did the ribosomes make in the rough ER? How deep's this rabbit hole go, Angela? How far can you get? You're all the way there. That's right. That's exactly where I wanted you. Now let's see if we can go further, right? So remember that there were different kinds of proteins in the cell that we produce, right? So the ribosomes that are out in the cytoplasm make all the proteins that just float around and bob around and do all their jobs. But the rough ER makes specialized kind of proteins, right? There's two kinds of proteins that the rough ER makes. Does anybody remember them? Give me one of the two. Go ahead, Alexis.
Secret sec. I can't pronounce the word.
It's okay.
Secretory.
Ready? Secretory proteins.
Yeah, secretory
They're
proteins.
secreted. Good. Okay, that's one. What's the other one? Angela, did that trigger your memory a little bit? There's something sitting. No. Okay, that's fine. Anybody?
Was it for like the membrane?
Okay, so Tyler said okay, technically you're right. And that's the best kind of correct. Glycoproteins are created by the rough ER. The glycosylation part. So if you guys remember, glycoproteins are proteins that have a sugar on them. They have a sugar moiety attached to them. And the attachment happens in the Golgi, right? Remember the Golgi apparatus. So what that glycoprotein, in order to do what it's doing there, what kind of protein is a glycoprotein with respect to membranes? April? You're answering my question. I know how you're like, I'm just going to say it out loud and see how it sounds. And if I like it, then I'll unmute myself. No, so I want to hear, what was your thought process there?
Well, I kind of saw what Lizzie put, and she said structure proteins. So I was thinking, I'm like, that kind of sounds right, like structural proteins.
Okay. So kind of. It's even more general than that. Remember, so there's soluble proteins in structure, or you're thinking of maybe the filament proteins like actin and stuff. Yeah. Okay. What about proteins that get actually put in the membrane? That's what we're going for here. Remember, I described that some of the proteins that the ribosome makes in the rough ER are stitched into the membrane, and I drew this. Remember this? Remember the stitching like that? We stitched the protein into the membrane. They're called integral membrane proteins.
Can you spell that?
Yes. It's on the slide. Let me just go to the slide. Just skip all this right now. I'm trying to teach you guys in a more active way rather than just mindlessly going through the PowerPoint here. Where's the word integral? Here we go. All right. So these are the kinds of proteins that interact with membranes. There's integral proteins. And what that means is the polypeptide chain is literally stitched into the membrane at the time it's made by the ribosome. So as the ribosome is going through translation and that polypeptide is getting extended, that polypeptides are getting directed into the membrane. Your book actually goes into some details there. That's beyond the scope. So that's why this study guide is useful. You don't have to know the level of detail that the book goes into there. Okay, so there's integral proteins, and then there's another class of protein called a peripheral protein. By peripheral, the English word peripheral means nearby. Okay, so like Jack right now, if he was standing literally right here, you can kind of see him, but kind of not. He'd be peripheral to the screen. Okay, just right next to. So a peripheral protein, to doodle that, if this is the membrane, an integral protein would thread through the membrane. A peripheral protein would just kind of like mushed into it a little bit, like a really comfy pillow and putting your head on it. Or like in Finding Nemo, the way clownfish interact with the anemones. They kind of like tuck in there. So you can that's a peripheral protein because they can easily come out again. Okay, integral proteins are permanently stitched into the membrane. You can't get them out. And once they're there, they're there. Okay. So let's talk just for a second about integral proteins here. Integral proteins have, so have I introduced this concept called a domain to you? Get a protein domain? Did we talk about those yet? Anybody remember? No? Okay, a protein domain is just a part of the protein. It's a functionally distinct part of the protein. We've learned that proteins have that primary structure. We fold it up and you get the various secondary structures. And then they have this overall 3D structure, that tertiary, right? And so when we consider the tertiary structure of a protein, we can see that protein as a machine that has different regions of it. Like, so think of your body right now. Everything's continuous, right? Your head is attached to your shoulders and your arms and your legs and your torso. So if I talked about your head, or I talked about your finger or your arm, right? You would call this your head domain, your leg domain, your elbow domain, or really your arm domain, right? So a domain is just a functionally distinct part of a protein. So if I have an integral membrane protein, also called a transmembrane protein, let's do this. Let's pretend it's got a doodle like this and like this and like this there. So I just drew a bunch of alpha helices and stuff, right? So this part of the protein here that's outside the cell, that is the extracellular domain. Okay, inside the cell's part is the intracellular domain. Okay, so here's my question to you guys. Which part of the protein would a hormone bind to? Or if this was a receptor on the cell's membrane, which part of the protein, which domain of the protein would be the part that binds to the important signaling molecule? The extracellular domain, right? The one on the outside, sure. Okay, and the middle part here, that's called the transmembrane domain. Remember, trans means across, right? Cis and trans isomers. The trans isomer is the one that's on the other side. The transmembrane domain goes through the membrane. It goes across the membrane. You can imagine if I took the primary sequence of this protein and we looked at the 40 amino acids that are making up that extracellular domain, we'd probably find polar amino acids and maybe even charged amino acids. And if we looked at that stretch of if we looked at that stretch of the sequence that goes across the membrane, we'd probably find a bunch of hydrophobic, non-charged, non-polar amino acids. And then we'd see another stretch of charged ones on the inside. And sure enough, when a scientist reads through a protein sequence, they're looking for stuff like that. They're looking for regions of hydrophobic and hydrophilic amino acids in there. And that's the telltale sign that you found an integral membrane protein. And some of these proteins will actually span the membrane, not just once like this, but they will literally stitch across it several times. And so when you just think of the sequence there, let's just earmark these, right? Those parts of the protein go across the membrane, right? If I pulled that apart and I looked at just the primary structure, I would see these alternating regions of hydrophobic, hydrophilic stretches of amino acid. Does that make sense?
I have a question.
Yeah, go ahead.
So is a protein either an integral protein or a peripheral, or can there be one that's like kind of both?
Oh, that's a great question. No, it's one or the other.
Okay.
Yeah. And then I guess the third kind of protein that we're not talking about would be one that's just like in the cell hanging out, right? And you can call that a soluble protein, I suppose. Here. So they're soluble, but really they're called globular. It's a fun word. They're globs, right? They're called globular proteins. So there's really, all proteins can be loosely classified into two kinds: filamentous and globular. Right? Basically, if you put it in water, what happens to it? Is it like albumin, like an egg white, and it's goopy, or is it like hair on the top of your water?
What's globular protein?
Yeah, so a globular protein is a protein that's soluble in solution. So cytosolic proteins, the proteins that are bopping around inside your cell, running your metabolism, those are all, like most of them are enzymes. Those are globular proteins. Okay, and if it's part of the cytoskeleton, if it's filament, those are filamentous proteins. Okay, so they're soluble or they're not soluble. That's the rough way of categorizing proteins. Okay, all right. So now that you understand that, let's talk about the ones let's go back to the membrane now. Okay, so here's my question again: how do we get something across that membrane that otherwise is impermeable to it? Like a reasonably sized protein, or hey, how about this? Glucose, right? You know that your cell needs to eat glucose, but glucose is too big to get across and it's polar. So how does glucose get into the cell?
The protein carries it.
Yeah, exactly. There's this whole world of proteins called protein carriers. There's carriers, so they're called carrier proteins, and there's things called channels. So there's channel proteins and there's carrier proteins. Now, the difference is the channel protein, think of the word channel, not in the sense of changing the channel on your TV, but a channel like the English channel, right? A pathway or a corridor through something. So a channel protein literally creates a hole in the membrane. And I can't draw today because remember this whole thing is broken. So that's why everything is like messy and weird. Let me just look at the PowerPoint here. There, right here. See that guy right there? That's a channel protein. It literally creates a tunnel for something to get through. They are specific to whatever's coming in. So there are things like chloride channels. That would be an example, a chloride channel. There's also the sodium-potassium pump. Right? So what do you think the difference is between me calling something a channel versus a pump?
A channel just allows it to go through on its like on a cord, and then a pump kind of pushes it through or out.
Excellent. That's a great way. So like when I say the word pump, you're thinking energy, right? There's that word energy we talked about earlier. So think about it. There are some things that we're okay flowing downhill, like flowing out down the concentration gradient. Like let's say you're building up a bunch of waste in the cell and you just open a hole and it just flows out, right? You don't have to worry about pumping anything because it's a higher concentration inside the cell. And so it's just going to flow downhill. Right? And then there are other things that there's not a lot of it in the environment. We want to grab it and bring it in against its concentration gradient. Right? And so that's the difference between the pump and the channel. Okay. Now, in addition, there are some things like, so the right-hand side there, that's the pump. You can see it's pumping red dots into the cell or whatever. But notice that molecule there, the one on the right, it's got like a weird shape to it. You see like there's like a wedge on the bottom and then a tunnel. There's like a different shapey thing there. So it's not represented in this picture very well, but what that's doing is that's changing its shape. It's literally changing its shape to pump things across. And so these kinds of things that change their shape are called carrier proteins. Okay, think about glucose again. You can't just open a hole in the cell and let glucose in because then everything's going to come in. Okay, so instead, you have this class of molecule that binds to something specific and then kind of moves it into it by changing its shape. Think of a very strange door that looks kind of like goopy jello, that when you go into it, it kind of wraps around your body, brings you in, and then puts you on the other side. And the whole time it's like changing shape. Okay, so there's carrier proteins and there's channel proteins. And some of those carrier proteins are classified as pumps if they're pumping things uphill and using energy. In this case, in this diagram, you can see the ATP there. Okay.
Can I make like a weird analogy real quick?
Yeah, please. I love weird
So-
analogies.
Have you ever seen Monsters vs. Aliens?
Yes.
So you know the little blob, like his name is Bob, and he will like eat things and then he'll kind of shoot them out if he can't actually die.
Yes. And that's kind of like phagocytosis. Yeah. So that's now the only problem with that, and my analogy too, my weird door analogy, is that it's more like a machine with moving parts, not a liquid gel. This isn't jelly. This is like my little toy here. This thing can change shape to move something through it, right? So this thing can move the pieces of it. It's like a machine. I got it. This is the classic example I usually use. I don't know why it fell out of my head. A turnstile. If you ever go to the subway or some big stadium, you have to go through the turnstile, right? The turnstile moves and it gets you through it. And they can also lock it so you can't get in, right? You just jump the turnstile. But that idea of a moving thing. Okay, good. All right,
Wait,
now the
what
rest of
about
this.
those?
Go ahead, Alexa.
Oh,
What?
sorry. One of those toys where it's like a tube and you can like you hold
Yes,
it and it like folds in on itself.
Yes. That would be another good example. Actually, that would be more like a channel though. It's got a hole in the center, right? So, but yeah, that comes to mind. I forgot about those things. Yeah, those are fun. That reminds me like Mobius strips and all that. Like that's like an infinite surface area somehow. Like, okay, good. Okay, you guys are getting this. So back to the back to the slide here. So ready? We got a cell. Think about this now. You have this cell here and you have this cell here. Okay, let's call them cell A and cell B. And if I told you that cell B can let some things in, but cell A can't. Why? What could be an example difference?
One's positive and one's negative. Like they don't let in polar or non-polar things.
Oh, I like that. Okay, no, but so in theory that could work, but remember there's one underlying rule here, and that is most cells, I always say most, like all cells to my knowledge, the inside has established a gradient using a sodium potassium pump, which I'll teach you how those work in a bit. Okay, so what else could be different between them? Why would cell B let something in that cell A is not letting in?
Like the different channels we just went over, like it would contain these different protein
Exactly.
substance.
Right? So cell A might be a skin cell and cell B might be a neuron. Right? So one of the things that makes cells different is they have different components to their membranes. This is the reason why why the SARS-CoV-2 virus can infect some cells but not other cells. It's also why, as we're learning, it can infect a lot more different kinds of cells than we thought. We used to think it was limited to the respiratory system, but once the virus gains passage into your body, it turns out there's a whole other slew of different tissues that it infects. And it turns out it's because the protein it's binding to, which is it's normally a receptor for, wow, it's been too long since I put walls up at this point for this stuff. Let's just say here, there's a protein in the membrane that, for the life of me, I can't remember the name of it. And it turns out that is found in a lot of tissues. There's a lot of different cells that express that. So the virus does its little secret handshake to that protein, and that's how it gets into the cell. I gotta look this up because my brain isn't failing me. ACE2, the ACE2, you guys have probably seen this in the news, the ACE2 receptor. The ACE2 receptor is involved in maintaining our blood pressure. And so it's found on all endothelial cells. It's found in a lot of different tissues, including our lung cells. And so once the virus gains entry to our lung cells and it spreads, it can infect the kidneys, the liver. It can even get into our nervous cell system. This is not the flu. The flu virus does not do that. Okay. And so the one little secret thing that I could start planting seeds for here is ultimately what's different between A and B is their genes, right? Even though these two cells have the same genome, cell B has some genes on that cell A doesn't. And that's what makes the cells different from each other, is we get to decide, or the cell gets to decide which genes are on and which genes are off. And that's what makes cells different from each other, even though they have the same genome. Everybody's working from the same cookbook. Okay. All right, great. All right, so is this working? I know I'm being more chaotic with the PowerPoint, but I'm being more interactive. Is this working for you guys? Like, yeah, I like this too. And for those of you who are introverts and like hate this, I'm sorry. There's always gonna be people that hate this kind of stuff. So I'll trade off every once in a while. All right, let's go back now and do a little bit more classic storytelling here. So we learned the rules of the road. This is the slide you can study. If you can recreate the slide from scratch from your head, you win. Okay, this is how the game is played. This isn't the chess game. This is how the pieces move, right? And your book does you a disservice, and I really don't understand why they don't put the oxygen and carbon dioxide in there. I added that. You can tell because they look like PowerPoint circles, because they're PowerPoint circles, right? But they should be there because the next 2 chapters are going to talk about these molecules they just go on and on about them. Right? We're going to learn about photosynthesis and cell respiration, and you should have an intuitive understanding of how those work. Okay. So now the last thing I want to talk about here is water. Water is a very interesting molecule in the sense of permeability, because for the longest time we used to think but couldn't explain well from a scientific perspective that water went across membranes very easily. And it does. We used to think it's because water just permeated through the cell. I'm sorry, water just diffused across the membrane. Like it just went across the membrane like oxygen or carbon dioxide. It just went a little slower because it was polar. However, that thought process could not explain why some membranes allowed water to go through way faster than it looked like it was supposed to by diffusion, and some membranes way slower. So I want you to picture a water balloon that's damp. It's just clammy. And you dry it off with a towel, and then within a couple seconds, it's just clammy and damp again. Right? So in other words, it's like the rubber's just a little leaky to water. And that's how I want you to think of cells. That's how they are. Cells are just a little bit leaky to water. They're not airtight. They're not watertight. They're not waterproof. Okay, without coatings, we can put coatings on them and do that. Like plants have a waxy cuticle that traps the water in there. So yes, the surface of a leaf, yes, but that's not a cell, right? That's a leaf. A cell itself, without a cell wall, without any of the stuff around it, just a membrane, is leaky. It's damp. Okay, there's a slow leak of water. So the question is, though, is some cells do very quickly let water come in and out. And it turns out there's a whole secret class of protein we discovered back in the 2000s called the aquaporins. Aqua, water, porins, pores, like pores in your skin. The aquaporins. And so that's why this picture here, you see this green molecule that the water is going across? That's an aquaporin. It is a water channel. It is specific to water. And this is what allows a lot of osmotic regulation, okay, which we'll learn about in this chapter too, osmosis. Where are we with time? Okay, good. All right, do you guys need a break? You guys want to take a break?
We only have like 10 minutes of class, so.
Oh, that's true. Oh, that's right. I'm sorry. In my head, it goes to 140 because that's my micro class. The other class goes to 140 on the other days. Yeah, poor time management skills. All right. So, okay, this you can use for studying. Like I could sit here and ask you, so we got something that's positively charged and polar and big, for example, some proteins. Can that get across the membrane by itself? Right? No, of course not. How about if it's negative and polar and big? No. How about if it's neutral and polar and big? No. Right, you guys are all in unison here. You guys get this now. That's good. Good, and that's what I wanted to take home to today to be, is the rules of the road. So that's good. All right, so the last 10 minutes here, I'm going to tell you the story of how we got to this point, how we understand all this. So the membrane itself, we call, and our best understanding of it now is called the fluid mosaic model. This is how we understand the membrane, and it's made out of lipids and proteins and carbs. And the lipids are phospholipids and cholesterol. The proteins are both the integral and the peripheral, and the carbohydrates are attached to the lipids and the proteins. So you got your glycoproteins and your glycolipids, right? So everything on this slide, everything makes sense. We've already covered all this. Okay, so for the rest of this lecture, what we'll pick up with on Wednesday is we're going to focus on these next 3 topics. Okay, so this is just the overview of the rest of the lecture. That's the earliest picture of a membrane. We didn't know it was a membrane at the time. This was taken in 1840. Somebody was looking under a microscope, and they were looking at muscles. They were comparing human muscle, and I think it was a lizard muscle or an alligator muscle. And they were finding, this is the period of time when biologists were realizing that the underlying structures of animals, even though they look different on the outside, right? There's not too much in common between an alligator and a human. At this level, it was like all the same. They were able to look at, like, muscle cells are muscle cells. right? So the building blocks of animals are like all the same. The building blocks of plants are all the same. This is beyond cell theory, the idea that all life is, made of cells. But what you're looking at there are two muscle cells connected by their cell membranes. So really, it's really one cell with two parts to it. But this little billowy part right here, that's membrane. That was in 1840. By 1895, this guy Overton realized that the membrane of a cell is made out of lipid. In other words, so he did these experiments. Chemistry was more advanced at the time. And so what he learned in his chemistry was that things that are hydrophobic, lipids, move across the membrane more readily than things that are hydrophilic, water soluble. So if you think of water soluble things and fat soluble things, the fat soluble things were able to go across the membrane. By 1920, this couple here, Gorder and Grendel, they did some really ingenious experiments to postulate that the membrane was a lipid bilayer without looking. This is how they did it. And we're going to end today on this story here. There's this device called a hemocytometer. Hemo for blood, cytometer for measuring cells. You can have basically it's something I use this in lab every day. This was like my hammer in the microscope lab. A hemocytometer it's made of glass, and it has this grid etched in it, like this. And from a side view, it looks like this. So the etchings are here. And what the idea is, you put your cells. So you take your cells, you mix them up into a solution so they're evenly distributed, and you quickly pipette them. You put a drop on the hemocytometer, and you put a cover slip on it. And then you look under the microscope. And when you're looking at under the microscope, you will see this grid. And you will see cells randomly on the microscope slide. And all you do is you count the cells within the grid. You see the bigger grid here? You can see there's a square here. You count the cells in that grid, and you say, I got 27. And you go somewhere else on the slide, you count, you count like 5 or 6 of them, and you get some average number, like 25.2. And then you do some math. You know that you added 1 microliter, you had, 10, you had 1,000 microliters, and so blah, blah, blah. There are 120 cells per milliliter, right? That's how we count cells. Okay. And so what Gorder and Grendel did was they took some red blood cells and they put it on this really cool device. I think I have a picture of it, actually. Yeah. I updated this last semester. So you see that weird membrane, that weird machine. What they did was they took the cells and they lysed them and they extracted their membrane. And then they let the membrane spread out on this plate, forming a monolayer, and they measured it. So in other words, they were able to, at the macro scale, take the surface area of the cell's membrane. Parallel to that, they knew how many cells they used, because they counted them under the microscope with the hemocytometer. They did some math, and this is where the brilliance comes in here, they assume the cells are spherical, right? So they took the formula for the surface area of a sphere, and they used that to calculate, they took the amount of lipid they had, they took the surface area, and they deduced that the amount of lipid they extracted from a given set of cells was always twice as much. That there was always enough lipid to make a double layer around a cell. Okay, and so knowing the chemistry of the phospholipid, knowing it was an amphipathic molecule, they deduced that you would get this double leaflet structure. And it wasn't until the 1940s, when electron microscopes were good enough, that they confirmed it. So they figured this out just by this brilliant math and extraction technique. Pretty cool. Okay, and That's where we'll end today. Okay, so what I want you guys to try to do, try to read through chapter 7 on your own. Do the dynamic study modules. Don't do the homework unless you want to, but really, I want you to try to do the dynamic study modules for chapter 7 on your own. Outline the chapter, go through it, look at my PowerPoint, look at the lecture notes, write them in your own words, build your own set of notes for chapter 7 using the study guide as a template or as a literal guide. Okay, and then I'll come in with, we'll go through this lecture, but I'm also gonna put the clicker questions in, okay? And we might have a quiz, we might not, I'm not sure yet. Okay, assume we will. And if we do have a quiz, it will be just on the basics of membrane permeability. We're not gonna go into, and the protein stuff. I taught you the protein stuff. So again, it's not linear in the PowerPoint. It's the topics we talked about today that you took notes on. Okay. All right, any questions? All right, so I'll yeah. Oh, what'd you say, Tori?
Sorry. So I noticed somebody said in the chat if there was any possible way that you could rename the recording in the Zoom.
Yeah, and the answer to that is unfortunately no. I have no ability to modify or tweak any of that. That's all Zoom's integration into Canvas. And I'm sorry, and it stinks. You have to basically click on them and figure out the method to their madness.
Okay.
So here's what I will say. There's four recordings. Okay. The first recording is just me, just this. That's useless. The second is just the class. That's useless. The third is just the PowerPoint. That's useless. The one you want is the one that has the PowerPoint and then me in the corner.
Okay.
That's the only one you should be watching. And so just click on it, scrub somewhere in the middle. And if you see PowerPoint and me, that's the one you want to watch. If it's not, just go on to the next one. I mean, you have a 25% chance to find it every time. And then I don't even think there's a method to it in terms of like the way they lay them out from recording to recording. I think it's in a random place each time. It's frustrating. And I'm sure Zoom's aware of this, and I'm sure they don't care right now.
There's no way for you to upload just the PowerPoint and you in the little corner, because like with my other classes, they don't have like 8 different recording things of the same thing.
Hold on first. Let me end this recording so you don't have to keep listening to this. Okay.